Good morning, online bio people. This is probably the second to last video you'll see from me this semester. I just wanted to spend some time going over the skeletal muscle packet and the neuromuscular junction packet, and then talking about what happens when an action potential travels through a muscle cell. So I posted the answers earlier this week on Moodle, and I think most of what you see in this packet is pretty self-explanatory. We've talked about how a muscle is kind of made of smaller parts that look like smaller parts that look like smaller parts that are all found in the muscle. So whenever you're looking at a muscle, you need to make sure you understand what you're looking at. So whenever you see tendon, you know you're looking at the whole muscle, and that the divisions are going to be fascicles. So surrounding the whole muscle is the epimysium, and surrounding the fascicle are the perimysium layers. So here we have a fascicle, and we know we have a fascicle because we don't see tendon, and we have muscle fibers with their nuclei on them. And so surrounding the muscle fibers, which are also known as muscle cells, is endomysium. And surrounding the fascicle, again, is perimysium. And now we're going to look into the muscle fiber itself. And the endomysium has been removed, so we can see the sarcolemma, which is just a fancy word for cell membrane in a muscle cell. We have our nucleus, and our myofibrils. Myofibrils have stacks of sarcomeres. Then remember, the sarcomeres are the contractile units. We'll look at those a little bit more in detail shortly. The other parts that we covered in lab is the sarcoplasmic reticulum, which is the endoplasmic reticulum of the muscle fiber, and the triads that have the terminal cisterna and the T-tubules. So we have two terminal cisterns, one from each side's sarcoplasmic reticulum, and that's where calcium ions are released into the cytosol. And going down, so here we have our sarcomere, our contractile unit, and so the Z lines on either side are where the thin filaments bind. Okay, the M lines connect thick filaments on either side. So those are the connection points. And then we have the dark band in the middle is called the A band. The dark band is where the thick filaments and the thin filaments overlap for the most part, except for this H zone in the middle, which is mostly thick filaments. And then we have the I band, which is mostly, almost exclusively, thin filaments. So the thin filaments make the light bands, and the thick filaments make the dark bands. And the slightly lighter region in the middle is where just thick filaments are found. And so here I will give you my way of remembering which filament is thin and which is thick. And if you are active, you are more likely to be thin. So actin is the thin filament. Okay, so I feel like we've probably done a good enough review here. All the answers to these questions are available on Moodle. So let's now move on to the neuromuscular junction. And so we have this worksheet. Again, answers are on Moodle. But I wanted to just go through the steps of action potential when you're talking about it on a muscle fiber. So we talked about action potentials, being generated by the axon hillock, traveling down the axon, having those voltage-gated ion channels opening, changing the voltage of the axon. And then when it gets down to the axon terminal, the voltage-gated calcium channels are going to open. Calcium's going to go rushing in and tell the synaptic vesicles at the axon terminal to release their neurotransmitter. So here we're talking about muscle cells, so we're going to be releasing acetylcholine, and the acetylcholine is released into the synaptic cleft, and that acetylcholine is going to drift throughout the cleft and eventually bind with ligand-gated channels on the motor end plate. Okay, so when the acetylcholine binds to the ligand-gated channels on the motor end plate, it's going to cause this influx of sodium into the cell, changing the membrane potential, and a small efflux of potassium happens as well. Okay, from there, we're going to have the that membrane change is going to travel down and travel down the sarcolemma. So this is another action potential traveling down the sarcolemma. And when it hits these T-tubules, that action potential is going to go deep within the cell. And going deep within the cell is going to trigger the release of calcium from the terminal cisternae. The calcium from the terminal cisternae is going to be in the cytosol. And it's ultimately going to be what causes contraction of the sarcomere. And that will be in the next video. So let me just go over that again. So the action potential travels down the axon. The calcium channels, voltage-gated calcium channels, are going to open, releasing telling the neurotransmitter to be released into the synaptic cleft. At the synaptic cleft, those acetylcholine molecules are going to bind to receptor sites. Those receptors are going to allow an influx of sodium and a small efflux of potassium, which is going to change the membrane potential of the sarcolemma. That change will ultimately lead to an action potential traveling down the sarcolemma of the muscle fiber. And that action potential will trigger the release of calcium from the terminal cisternae. One more thing I want to talk about before we go is that acetylcholine is going to release from its receptors, and it's going to be broken down by something called acetylcholine esterase. And the acetylcholine esterase breaks it down into smaller parts that can be taken back up into the axon terminal and reformed into acetylcholine. I hope this video is helpful, and please feel free to send me email over the weekend with any questions you have. Thank you.